In this 3-mark subjective question, they are directing us to explain the DNA replication mechanism with the help of replication fork, and they are directing us to specify in which way the DNA ligase enzyme is working in the DNA replication fork. For this, we are specifying the DNA replication starts from the specific site called origin of replication. For the long DNA molecule, since the two strands of the DNA cannot be separated in its entire length due to the very high energy requirement phase, the replication occurs within a small opening of the DNA helix, referring to the DNA replication fork. For this illustration, we are getting word of mark. The DNA dependent DNA polymerase catalyzes polymerization only in one direction, that is 5' to 3' direction. This creates some additional complication at the replication fork. The consequences on the other strand, the template with the polarity 3' to 5', the replication is continuous, and the other one is indicated in the discontinuous strand synthesis. The discontinuous strand synthesis fragments are later joined by the enzyme DNA ligase enzyme. For this specification, we are getting overall 1.5 mark for the illustration. In this way, we are satisfying the 3-mark demand of this first optional subjective question. In this 3-mark subjective question, they are directing us to construct and label a transcription unit from which the RNA segment given below has been transcribed, and they are directing us to give a complete name of the enzyme that transcribed this RNA. For this, we are giving the detailed double helical DNA structure with the transcription components. In this particular picture, we can see the promoter is a landing platform for the RNA polymerase. Once RNA polymerase is landing, immediately unwinding of the double-stranded DNA happens. That facilitates the RNA polymerase to work more effectively to synthesize RNA strand only with one strand, not with the other strand. While encountering the terminator, the RNA polymerase gets away from the unwinded DNA strand. With this specification, let me illustrate the sense strand in the RNA in which way it exists. For this, we are giving the specification. For this, we are getting half mark, and for the diagram illustration, we are getting 1 mark. And for representing the antisense strand, for this corresponding strand, we are getting another half mark. So in this way, we are satisfying the 2 mark. For specifying the complete name of the enzyme involved in this transcription process, we are illustrating the process of formation of RNA from DNA in this transcription process is because of the enzyme DNA dependent RNA polymerase or RNA polymerase. This gives another 1 mark. In this way, we are satisfying the overall 3-mark demand of the second optional subjective question.